This video is going to take us through Entrez and just give an overview of how to use it. So if you would Google NCBI Entrez, this is the page that you would most likely come upon. And we'll go through this page and then we'll actually go to the query page. But I wanted to show you this because it gives you kind of an overview. So the first thing to notice are the Entrez databases up here. And the things that you'll have access to, and these are just a few of the things, are nucleotide sequences, protein sequences, 3D structures, genomes, taxonomy, PopSet, which they're aligned sequences that are submitted as a set resulting from the population or phylogenetic or mutation study describing some event in evolution or population variation. And the nice thing about this is that it includes nucleotide and protein sequence data. And last on this list is PubMed, which I'm sure you've all heard of. And there are different ways to search, but one way is with text searching. And you can search fields within the database, but the fields vary depending on the specific database. Some of them are common to all of the Entrez databases, such as accession number, author name, text word, journal name, publication date. And other fields are present in most of the Entrez databases, such as a title word. And then the fields that are particularly useful in the searches are organism, properties, sequence length, feature key, and molecular weight. So if we look at the properties search field, this is very popular for protein sequence databases, and it allows you to limit your searches by a number of different things. So imagine you're searching the protein sequence database and you want to look for a particular molecule type. You can do that in properties. You can also use GenBank division, such as the ESTs or WGSs, and you can also use gene location or the source database. Like when we'll get into this, like the ref-seq records, you can sift out, or using Swiss prot records. And it's easy to use this in the limits page, which we'll also get to. And the other thing that I just wanted to go over a little bit from this page is that you basically have 3 levels of search complexity. And the basic search is just using keywords. And for instance here, you could enter cystic fibrosis human, and that will give you a multitude of results. The next level is referred to as advanced. So in this type of search, you can use the limits or index or history options. And just to give you an example, if you use limits, you could select the limits option, you could enter cystic fibrosis, and then you could also use title word so that you would want to have cystic fibrosis in the title. And then you just press go to use those limits in your search. Or you could use human as the organism and in the search field. And you can also use history, and this is based on your own search history. So you'll have to, after you make an account with NCBI, it'll keep track of what your searches are. So if you want to search for cystic fibrosis in one search and then search for, say hemoglobin in a second search, you could add those search histories together to see what you come up with. And the last one is complex Boolean searches, and this is where you use Boolean terms like and, or, not, but you can also use identifiers such as down here where it says cystic fibrosis title and human organism. So that's just another way to limit your searches. So if we scroll back up and we go to Entrez, click on Entrez, that should take us, oh no, not that Entrez, you have to click on this one up here. It's working a little bit slowly. This will take us to what's referred to as the gQuery Global Cross Database. So as you can see, there are a lot of categories here. So we have PubMed, which we mentioned before. We have the Online Mendelian Inheritance in Man. We have that one. And down here in genomes, we have a lot of different categories. There's the actual genome database, there's nucleotide, there's probes, there's SNPs or SNPs, and then you also have a genes category where you have ESTs, you have genes, you have homologene, you have unigene, and proteins, you have conserved domains, protein. So there's a multitude of different categories here that will come up with numbers when you search for something. So let's just search for something that's we know we're going to get a lot of results for. So you type in hemoglobin, hit return or click on the search button, and you can see how many results you get for each of these. So if we just go ahead and let's look at gene, for instance. If we click on gene, there's almost 5,500 results. Way too many to look at, but for the sake of example, they start out here with Arabidopsis, and then you go down here to the human species. You have all the different subunits. So we'll just click on HBB, which is hemoglobin subunit beta, and to show you what the gene record looks like. So here we are with the gene record. Gives you the name up here, gives you the gene ID. It was last updated August 27th, 2017. And it gives you, first it gives you a summary, gives you the official symbol, the full name, the primary source, gives you related sequences, and these are in different databases which we'll look at later also. So this is from Ensemble, this is from OMIM, and this is a Vega sequence. And it gives you other information. And it also gives you a summary of what the gene is. Then if you scroll down, it'll give you a genomic context. And I'll also point out you can, with this drop down arrow here, you can either hide things or view them just by clicking on that arrow. So this part, the genomic context, it gives you the location. So this is chromosome 11, P15.4. So it gives you the chromosome and the exact location. And then it gives you the exon count. And this gives you assembly information, like what assembly, like genomic sequencing assembly it came from. And this location number over here is the actual accession number for the sequence. And this just gives you a picture of the chromosome and where it's located. So down here you see HBB, and the arrows tell you in which direction the gene is transcribed. So in this case it's going from right to left. And actually all of these are that are here. And then if you go down further, you have genomic regions, transcripts, and products, which give you more graphics. So you have the genes here, and as you can see, things pop up when you scroll over them. So this is giving you information about the actual gene location and what base pair the gene is located at. And then you have this is NCBI, this is Ensemble. You have SNPs, which are all these little dotted lines along here. So each one of those hash marks is a SNP. And you have clinical variations, you have cited variants, and these are also SNP related. You have RNA sequence information, and that obviously gives you exon intron information. And as you see up here, you have features up here where you can manipulate the picture that you're looking at. So you can move either left or right. You can zoom out or zoom in, and you can zoom to a specific sequence if you know a sequence, and you can switch on the different modes for the gene tracks. And you also have different tools, and this is just a list of the tools. So going beyond that, it'll give you expression information, and there are links to journal articles here, and then it'll also give you a bibliography at the end. So quite a lot of information. And also down here we have phenotypes, and this gets into disease-related things. So I mean, and you have to remember that not all of the genes will have all this information, but since this is hemoglobin, it's going to have a huge amount of information. So we're about halfway through the page. So we have variations, and these have to do with SNPs again, if you look at genotypes. And then you have pathways for biosystems. So this gives you pathways for like receptor activation, anything that might be disease related, and where the actual mechanism is where the disease occurs. And then you have interactions, and this gives you the product, and it also gives you the interactant. So this is important if you're looking for maybe protein-protein interactions or other genes interacting in some way. And then we get down to general information, and one that is particularly important are markers. So if you click on, you noticed I clicked on that, and this gives you all the markers for this particular gene. A lot of STSs, ESTs probably in there, and a lot of these are in the UniSTS database. And these are also related to SNPs, so I'm going to minimize that again. And all of these lists, you see that's either plus or minus. You can minimize or maximize what you're seeing. And then there's a list of gene ontology, and then general protein information, and then we get down here to the NCBI ref sequences, which they're most highly annotated. So you have the ref gene sequence. This is mRNA, this is protein. So if you want those sequences, you can readily download those and work with them. And then there's also related sequences, and these are typically sequences that the ref came from. So they've been annotated and verified in some way. Then at the very bottom you have additional links. So a lot of information on Entrez gene. So if we go back, another one that I'd wanted to show you is just OMIM real quick. Taking a little bit to go back. All right. To go back 2 steps, I think. Here we go. Okay, so we looked at gene where we had 5,400 possibilities. Let's go to OMIM, and here we have 249. But this is more disease related, and it'll show you. See, this is the list of the 249 results. Let's see if we can find. I don't see HBB off the top here. Where's HBA? Here we go. HBB. Let's take a look at that one, and you can see that it's the same chromosomal location. If you look here, the 11p 15.4. So if we click on that link, it'll take us to the OMIM page. Eventually. Here we go. Okay, so just a few tips about this page. This, like I said, it's more related to disease. So this is your chromosome location again here, and I'll close that little ad there. It gives you a phenotype. So one of the more common ones is probably thalassemia. So the first one is delta beta thalassemia, and that's the phenotype, and it gives you a specific MIM number or a MIM number, and it does that with most of these, not all of them. So if you scroll down, I mean, if you know what you're interested in right off the bat, you can click on one of these MIM numbers. But if you just want to find out more about HBB and the diseases it might be involved in, this OMIM gives you a lot of detailed information. So it'll first off give you a description, and again you can minimize these things or expand. It'll give you a description, it'll give you the gene structure, and it'll give you good references as far as when it was discovered, and then it'll tell you a lot about the mapping. So how did they map that gene to chromosome 11 and P15.4, and it'll give you any pseudogenes that might be present, and then it goes into gene function, which is, you know, like I said, a lot of great information in OMIM if you're interested in any diseases. And then you get into biochemical features. Here they have a paragraph on crystal structure, and then molecular genetics. And here it gets into the diseases. And they have the beta, describes the beta thalassemias, and again, who discovered them originally, how they were mapped, what descent of or ethnic background they're typically found in. So a lot of information, and here's, I mean, the beta thalassemias, they are probably would have the most information, but you also have erythrocytosis, the hereditary persistence of fetal hemoglobin, delta beta thalassemia, protection against malaria, and then it gives you a whole list of reviews, which again is great information. So if let's see if we can just see all the categories here. There's a lot of information to scroll through. If you see the sidebar there, we're at the very top of the information. So let's minimize some of this so we can get a better handle on everything that's provided. Okay, so next category here is animal model, and these are all over here too if you want to jump to just one thing. So it tells you all about the animal models that have been used, it gives you the history, and then it also takes you through each individual allelic variant, which is super for bioinformatics because it tells you specifically where the SNP is. It gives you a reference number for the SNP, so you can go directly to the database and find it. And then the last here is other references to look at. So OMIM gives a lot of great information if you want to look into diseases. So hopefully that's given you a pretty good background of the Entrez database and how to access it. And I think that's it for now. And we'll take another look at how to use the limits in another video.